Today we are joined by Dr. Charles Evans, a Research Associate Professor at the University of Michigan and a member of the Michigan Regional Comprehensive Metabolomics Resource Corps. Dr. Evans, what makes the metabolomics of tissue samples so important?
Well, the goal of any metabolomics study, as you know, is to capture an accurate portrait of the metabolome of the entity that you're studying, whether it be a bacterial culture, a plant, an animal, or a human being. And the metabolome is very dynamic, and you want to capture any changes in the metabolome that occur in the system that you're studying. So in a higher organism, the tissue is the site that those disruptions may occur in, for instance, a disease or some sort of an experimental manipulation that you're performing. So it's important to not just get the average state, but also to be able to look at the actual site where the metabolome is altered. And that's sort of the basis for performing tissue metabolomics experiments.
What are some of the characteristics that set tissue samples apart from other samples used in metabolomics?
Sure. So classically, a lot of studies in metabolomics have been performed using biofluids. Things like blood or plasma or urine are very commonly studied, and that makes sense because they're very easy to access, and they also are a great way of understanding the average biological state of the organism that you're studying. However, in disease and in other conditions, as I've already mentioned, disruptions in the metabolome occur at the site of tissues, particularly if you have a disease that affects the liver is a logical place to look. So, tissue metabolomics gives you a more focused view of the metabolome and adds a lot to that picture that you would gain by also looking at the average metabolome in a biofluid. So that's one reason why it's important.
What are some of the most important considerations for tissue collection for metabolomics in animals, like rats and mice?
Sure. So the metabolome is an incredibly dynamic entity. Just a few seconds of alteration caused by something that you might do to collect tissues, like performing surgery or a cessation of blood flow, is enough to potentially induce very substantial alterations in the tissue metabolome. So these are things we need to be aware of in a tissue metabolomics experiment. One of the biggest areas of concern is in an animal study is the mode of anesthesia that you're going to use prior to collection of the tissues. We found that procedures that may work fine for other studies, such as genomics or proteomics or classical biochemical studies, may not work very well at all for metabolomics, and that's something that we found that it's very important to be aware of.
So you mentioned anesthesia. What are some of the effects of anesthesia on the metabolome?
Sure. So most procedures involving anesthesia ultimately involve sacrifice of the animal from which the tissues are being collected. But one thing that we've noticed is that there's a critical distinction between procedures where tissues are collected while the animal is under anesthesia but still breathing, compared to procedures which are performed in which tissues are collected after the animal has been euthanized, and tissue collection is then performed immediately thereafter. The metabolome, not surprisingly perhaps, undergoes a fairly rapid and dramatic alteration following euthanasia, and also there can be more subtle alterations that occur under different modes of anesthesia. For instance, one observation that we've seen in euthanized animals, we detected threefold higher levels of the glycolytic intermediate glucose 6-phosphate in animals that were euthanized, even though tissue collection was performed very rapidly following euthanasia, compared to animals where tissue collection was performed while they were under anesthesia. We also detected many other changes in the metabolome, not limited to skeletal muscle, but also present in many other tissues and many other classes of metabolites. You might think that this is sort of an obvious question, that obviously you don't want to euthanize your animal before you try to collect tissue for metabolomics, but perhaps surprisingly, there are a lot of studies out there in the literature that that's exactly what's done. And so we don't want to discount the data in all of those studies. Very useful and valuable data can be obtained regardless of the method of anesthesia that's used. But if the goal is to be as close as possible to the in vivo metabolism, that's what you want to study, then we prefer tissue collection procedures under anesthesia instead of post-euthanasia.
Can you recommend a procedure for tissue collection and for metabolomics in rodents?
Sure. So it's worth pointing out that no single procedure is going to be ideal for all studies. You should carefully consider your procedure and design it for the study that you're performing. And also, the next most important thing to point out is that any study that's going to be performed on animals involving tissue collection or any other procedure needs to be compliant with all national and international ethical standards for treatment of animals. and they also need to be approved by an authorized animal care and use committee. And so after that essential step is satisfied, the first thing that you need to do in order to collect tissues for metabolomics is to induce anesthesia. And as we've said before, our preferred method is to use a calibrated anesthetic delivery machine for some sort of an inhaled anesthetic like isoflurane. This allows rapid induction of anesthesia and maintenance of a steady level of anesthesia throughout the procedure. Once that's done, the next step is to begin your tissue collection. After you've incised into the animal to isolate your tissue of interest, you have a couple of options for how the tissue can be collected. The fastest and in some ways best method is to use a freeze clamp. In other words, liquid nitrogen chilled tongs directly clamped on the tissue of interest to freeze it in situ and then carefully excise the frozen piece of tissue. This is good because it's fast. The downside is that you can't very carefully dissect the complete piece of tissue that you may want. The alternative is to collect the tissue, surgically collect the tissue first and then freeze it by either freeze clamp or by immersion in liquid nitrogen. That's good because you get a more accurate dissection. You can capture the whole tissue that you're interested in studying very precisely. The downside is that it's slower, and there's a few seconds between removing the tissue from the animal and when it gets frozen, where alterations in the metabolome can occur. Okay, so once you've collected your tissue, it's worth pointing out that for procedures where you're collecting multiple tissues, it may be worthwhile to collect the peripheral tissues like skeletal muscle first, and then move to the more central tissues like liver or the heart in order to maintain normal blood circulation for as long as possible while your tissues are being collected. But regardless, if you have a procedure where the central tissues are most important to your study, then I would recommend that you begin with the most important tissues first, collect the liver, and then move on to tissues that are of lesser importance later. Once all the tissues that you're interested in have been collected, you can end the procedure according to your approved animal protocol. Store the tissue samples at -80 degrees C or colder. For sample preparation steps that come next, see the Metabolomics Core website. We have a video on that topic, or consult with the Metabolomics Core.
Can you give us any recommendations regarding the collection of human tissue samples for metabolomics?
Yeah, so human studies are obviously going to be a bit more constrained than studies performed in animals. It's essential that sterility be maintained and that minimal damage be induced to surrounding tissues. So obviously all of these things come with collection. Typically human samples are accessed by biopsy and very occasionally during surgery in collaboration with a surgeon. So there's a lot of constraints that are just imposed by that process in and of itself. But beyond that, the general recommendations would be the same as what we have in animals. It's beneficial to freeze the tissue as soon as possible after it's removed from the subject that you're studying, so by immersion or in liquid nitrogen or by freeze clamping. And it's also important to have very consistent timing from subject to subject. You want to be sure that you design the experiment in such a way that you can collect your samples quickly and be able to follow a very, very consistent and very fast time course in all of your subjects so that you can get consistent data from one individual to the next.
Do you have any other recommendations for our viewers?
Sure. In general, the last thing I would say is just no two studies are alike. It requires careful planning to design and implement a successful tissue metabolomics experiment. And I would recommend that our listeners consult with the Metabolomics Core in order to get help in designing the best possible tissue metabolomics experiment and get good data out of their study.
Thank you, Dr. Evans, for your time and for your recommendations. Dr. Evans and other experts in metabolomics can be contacted at the Michigan Regional Comprehensive Metabolomics Resource Corps. The contact information for the Corps is available in the video details. Thank you.